So mutations, but after use it, bacteria we have when we discharge some biological waste that contains also that bacteria, and all these bacteria are not very friendly. So some are pathogens, some are gram negative. So you know this better than me. So that can create, that can spread, that can even get some nutrients from the waste or from the soils, and that can spread, that can create the problem. So the we have to follow a strict regulation while we discharge that biohazard or biological waste. Similarly, whenever some chemical industries are there, they produce the waste material, so they discharge it. They should follow the strict rule that while you discharge, you have to discharge the waste material in such a form that nature should consume it or nature should tolerate it, because that should be the cycle. In our life cycle, what we follow, we are exploring, we are collecting the resources from the nature, we are transforming it to the useful products using the machineries, using the technology. After using it, we have to dispose it, and we have to dispose in such a form nature should consume it. Then only the cycle will complete. So if we are unable to dispose in that form where nature do not consume it, so what will happen? Gradually, so that waste material will gather, and waste material one day will become the harmful for the human civilization itself. Okay, guys, and I hope you even today or in the present day you understand the problem with the plastics, isn't that? The plastics, because plastics do not decompose. But if you use, guys, the paper decomposes with the help of bacteria itself, microorganism itself. When we die, what will happen? When any animal die, when plant die, so many of the so with the help of bacteria, so everything will decompose. So we vanish in this nature. So this vanishes means the nature consume us, consume all the material we made of. So here, the similar way, in the chemical industry, whatever we produce, it is our role, it is the role of chemical engineer to discharge in such a way that after treating in such a way that nature should consume it. And at the end point of consumption, we have no other option without taking the help of microorganisms. So now, guys, you understand. If someone asks you to deny to involve you in a job in the chemical industry, I hope you can defend. Okay, I hope you can defend and you can have a good debate with that. If as a chemical engineer I say, where will be your role in the chemical industry? I hope you can defend yourself. That the point you have to play. If I truly concerned about the environment, chemical engineer or chemical industry must adopt the biotechnologies or microbiological people with the biological expertise, because at any point, at the end point, the effluent, what I discharge, so that need to be consumed by the nature with the help of microorganisms or with the help of some biological process. Okay. So here you see, if I say anatomy of chemical process. Okay. So what happened, guys? Anatomy of chemical process. I told you, probably you don't have the idea that actually what are the steps or what are the units in the industry or any in a big plant. You can have a confusion over that. Big plants are there. So that is not the understanding of chemical engineers only. Guys also must understand that the steps. So in your language, if I say anatomy of a chemical engineering, So what steps you can say? The raw materials, the resources. We have to collect it either from the natural sources, maybe from underground or the earth crust, or from the surface. If there are many, you can imagine the different type of industry where the different material, raw material we get. So raw material storage, collection, and we have to store it. After that, the feed preparation will be the second stage, and this anatomy is true for all industries. Okay, feed preparation. What is the feed preparation? Some even you can have a good example from your house, from your kitchen itself. You want to prepare rice. You know, the washing of raw material here in industry is required. Sometimes washing requires, sometimes grinding requires. So we have to make the fine dust of that. Sometimes preheating required. So that is the feed preparation. That means we have to prepare the feed in such a way that the reaction could take place properly. So before sending the reaction or reactor, we have to prepare the feed. Even you can compare with the fermenter or biological process. So before sending the raw material into the fermenter need to be washed and sterilized to disinfect or to free from the other bacteria. We have to sterilize it. So sterilization is the feed preparation step. Sometimes we need to grind it. Sometimes we have to make a solution in it. Sometimes mixing, preheating. I hope you understand. So that feed preparation is the stage three. And stage three, then what to do? We have to send the prepared or pre-treated feed into the reactor. So in a true chemical plant, we have to send it a chemical reactor. In a biochemical process, we have to send into a bioreactor. And the difference between a chemical reactor and a bioreactor is that the reactor which involves the microorganisms or a bacteria or a cell, then only we can say this is. So any reactor where a microorganism or cell is playing its role, then we say term as that reactor as a bioreactor. So this is the difference. Only difference I hope you can find. Now we have to think that how they proceed or how they perform. So that we will learn one after another. A chemical reactor, because I hope you have the idea about the definition of the batch process and continuous process, guys. Batch process and continuous process, because I know that most of you are from you have to do the batch process, continuous process. So I want to listen from one of you. Okay. Sanjukta, can you explain the continuous process so that your friend can listen from you? You switch on your mic, then you tell, and then switch off. Sanjukta or Annesa or
Yes, sir.
Sandy,
Yes.
anyone? Yes, Sanjukta.
So, in continuous process, continuously the inoculum is added and the medium is provided in a continuous manner. But in batch culture, batch after batch, it is provided.
Okay, so suppose I have a debate with you. So my question, continuous inoculation, you are adding, okay, inoculate it, and the reaction, continuously you can get the output of that. So the rate of input and rate of output will be same,
Yes, sir.
should same.
Not exactly same. Like the depending on the log phase, lag phase of the bacteria, it may vary. Different phases of the growth.
Okay, so then what happened is gradually the amount of in the reactor will increases. amount of mass will increase in the reactor. I hope
Yes, sir.
I am deliberately confusing you, nothing more than that. I have no intention to catch your mind. I am confusing you because we people need to confuse ourselves with the process first. We have to find the fault in our knowledge, in our conception first. Then, so I know most of you, even I do not receive the exact answer from the chemical engineering student in the other batch also, because we imagine that continuous input, so continuous output, so it's a continuous process. Then batch process, we have to add at a time. Then after some time, we are taking all this material out of the reactor. And that time is called the batch time. Okay. And in continuous process, but there are many questions will come in the continuous process. The concepts like what should be the rate of input, what should be the rate of output, like that. So you are saying that may not be same. Someone can say yes, it should be same. Otherwise, regeneration will be there. So the exact definition probably eliminate all this confusion, guys. Is that a continuous process is a process where no momentary stoppages are there. Okay, then probably the questions will not come. Counter questions will not come. So we define the continuous process in such a way that is a process where no momentary stoppages are there. Okay. So anyway, I'll be discussing why I told you this definition later on. So when we go deep inside of our subject. So initially, I'm trying to give the outlook of this subject. So first, then now come to the point, anatomy of the chemical process. So we reach up to the stage 3 now, reactors. So it is the either in a chemical process is a chemical reactor, in the biochemical process I told you it's a bioreactor. So from the react after the reactor, the product will come. Products, maybe we have some targeted products, and obviously there are some unconverted reactants will be there, because most of the chemical process we find 100% conversion is not possible, and where you can have a debate with the chemical engineer and you feel superiority, guys. Yes, this is the point. You should feel superiority because you deal with the enzyme, or the because ultimately bacteria work through its enzyme. Okay, so the enzyme has the ability to convert, or acts as a catalyst, and that has very high conversion rate, even 99% or 100%, because enzyme is the selectivity and the specificity is very high. You guys work with the enzyme, and we chemical engineer treat or compare enzyme as enzyme with the catalyst. And we are limited here. We know the enzyme in our body, in our the animal being in any cells are specific, 100% specific. They are able to do a very selective reaction, and even 100% conversion could possible. And we chemical engineer, while develop the different type of catalyst composition, we always take the reference of the enzyme, and we always try to reach up to the performance of the enzyme, because nature, this is the natural enzyme, natural. Here we engineers lagging behind the nature because we could not reach up to the nature, natural creation. Okay, guys. So anyway, many products will form, and some unconverted obviously reactants are there, along with some byproducts, some unwanted products are there, whether it is fermentation, whether it is any chemical reaction, whether other decomposition processes, whether it is dark fermentation, whether it is photo fermentation, whatever. Okay, so always some byproducts are there. Okay, so that now you perform you perform the reaction, you select the bacteria to produce some useful products. Now, after the reactor, the useful products mixed with the unreacted, unconverted reactants and byproducts. So now, stage 4, what we have to do? We have to separate all these components very efficiently. Okay, so one of the very important area or important subject for the chemical engineers are separation technology. And do you believe that separation technology only pioneers for chemical engineers? I feel no. I feel no. This separation techniques or technology should be should learn by the people you are like peoples like you and students like you. Okay, because separation is also your part, and modern separation technique teaches in the chemical engineering branches. You know what techniques we are teaching to the chemical engineering students and the people, because this is a modern technique, and probably you are very much useful with this technique. That's is the very useful technique for you guys. So your technique we have considered as the modern technique. What is that? Chromatographic separation, HPLC, high performance liquid chromatography. So that chromatographic separation nowadays we chemical engineering trying to adopt and scale up so that we can use it industrially. Why? Because this chromatographic separation and the high performance liquid chromatography, as I say, high HPLC, thin layered chromatography, all type of that, the membrane separation. So this process has very high performance, high yield. Separation efficiency is very good. Okay, we can reach up to 99%, even more than 99%. I am giving you one example. Guys, the alcohol and water mixture. If you want to prepare 100% alcohol, how it is possible? I hope you know it. Suppose from 25% alcohol solution with the water, and we need to produce it 100% alcohol. How it is possible? Yeah, yes, it is the distillation. So using this distillation, because distillation generally used while the relative volatility or the boiling point differences are there, or we say relative volatility is there, difference is there. So now how much concentration or how much purification is possible by distillation, because distillation is the elevated temperature separation. We have to separate it using high temperature. So how much concentration we can achieve through distillation? Anyone? Yeah, Saurabh, do you have idea that how much purification is possible? Annesa, those
Sir,
are my
95% is possible, 95 to 96.
why
Um,
95 to 96. Okay, 4.6, because that form at that point what happened? Alcohol form azeotrope with water. Am I right?
yes, sir,
Azeotrope.
azeotropic means.
Yeah, yeah. Now you remind. So azeotrope will form. What does it mean by? Because we learn it in school level. So azeotrope means, in other words, it's a constant boiling mixture. That means the
It,
boiling point will be constant for that mixture. And molecularly, what actually happened? At that concentration, one uh, ethyl alcohol molecule becomes surrounded by 16 H2O molecule, water molecule, and that becomes a inseparable. So that form a constant boiling mixture. And I told you that if there are the difference in the boiling point or relative volatility differences there, then only distillation will work. Other than whenever this will be same, 1 or constant boiling point, then it is not possible by a simple distillation. Is not possible. Then how we can reach, how we can get the 100% of ethyl alcohol, guys? Any idea? Any idea? Okay. So I'm telling that it is possible by some special kind of distillation. It is called azeotropic distillation or extractive distillation. Third component we have to add, that we will mix with the alcohol or water, and what happens, that creates the difference or that break that bond, what I told you. A ethyl alcohol molecule generally surrounded by 16 water molecules, so they will be separated. Further separation is possible, but that becomes complicated because again third component contaminates the alcohol. So what should be the modern technique? We can use some membrane through pervaporation. I hope you heard this separation technique: pervaporation, permeation and vaporization. Permeation and vaporization. Permeation and vaporization. Composite pervaporation. So using some membrane it is possible. Okay. Or some liquid chromatography if there. So yes, we can think like that. Through the liquid chromatography we can separate it, because the property of alcohol and water are different. Those similarities as they are polar, still they have some dissimilarities. So we have to use that property of dissimilarities, then we can separate it. So like this, we have to think the separation technique, and chemical engineers also thinks different techniques like adsorption, okay, extraction, vaporization, evaporation. So there are different separation techniques are there. If the solid-solid mixtures are there, your products are solids, different type of solids. So separation technique will be, will be different. You have to think in otherwise, either by screening, either by magnetic separation, so that if some materials are ferromagnetic in nature, so you pass it through the magnetic field, so some will attach, some will not. So different technique we can say: homogeneous liquid mixtures, heterogeneous liquid mixtures. So that means if the liquid mixtures are there and all these liquid form the homogeneous solution, so the separation technique will be different. If some are heterogeneous mixtures, some have different phase, then we can separation technique will be something different. So, the product separation is a huge challenge. It is not only challenge for the chemical engineers, but also for the people involved in the biological or working with the biological, because you have to prepare the product, you have to separate the cell, separate the cell by centrifuge, using centrifuge. But the other products, acids, liquids, the alcohols, so you have to separate. Why separate? I'm telling some point. Why separation is very much important for you, that you will understand. The reaction takes place if you are able to immediately separate the products. So rate of reaction will be a constant, or you will be able to maintain the rate of reaction. Otherwise, what happens? Or if the product concentration increases, gradually you are not separating products from the reactors, and what will happen? The products will decrease the rate of fermentation or rate of bacterial activity even. And probably you guys are habituated with the term, it is called product inhibition. We chemical engineers are not habituated. We call the product concentration. So the rate, and through the some principle, La Satellar principle, we analyze. But you guys know that product inhibition. There are few terms is new to the chemical engineers, are called the substrate inhibition, product inhibition. Okay. So you are very much useful. Anyway, so product separation is very important. Okay. So now separating this product, if unconverted raw materials are there, you should not throw it out because that will be a wastage of money. So you have to think to recycle it and mix it with the feed and again send it to the reaction. If you find some byproducts and that are not required for you, but still engineering mindset, if you have, you never think that byproducts, okay, also this are it. No, no, no. You should survey the market or you should analyze the scope of use of that byproduct because you should have a thinking or mindset to earn money from the byproducts. It is not that you say this is not useful, this is not required by me, so you discharge it. I am giving you one example, guys. In your fermentation, you separate after the reaction. What will happen? You disinfect the bacteria or cell, and then what happen? The cell mass. What you do with the cell mass, guys? Anyone? Cell mass. You dispose it.
Hello, sir. No, sir, we can produce single cell proteins
Yeah,
like
single.
ACP.
Right, right, right. Okay. Thank you, Anessa. So, cell mass after separating, just you are thinking in your lab level. I am dreaming in a large fermenter in industry and separating a tons of cell mass. Okay, so cell mass, what we can
Yes,
do?
sir.
Yeah, we can
Sir, we
convert
can
that.
produce
Okay,
forestries
okay,
also.
okay. Please listen. So that cell mass, we can produce that some amino acids, some proteins from that cell mass. And nowadays, that amino acid we produce is from cell mass. We can use that amino acid, the biofertilizer itself, because whatever fertilizer I am using for the plant or designing for the plant, ultimately in the form of amino acid they are receiving, absorbing by the cell of plant cell itself. So amino acid, blended food, as well as the fertilizers is getting importance in the market. There are lot of biomass is available, biofertilizers available and coming in the market. Those are based on the amino acids, and that amino acid you could extract it from the cell mass. So that is my, point that byproduct you have to find out the scope to earn money so that you can send it to the other units before disposing of. Now, some waste material you find no uses. So suppose some gases are coming out. So if because ultimately the engineering point of view you have to think if that material you are using is not profitable, then no one will be users. So some can be, you have to minimize the waste disposal. So after that, so I hope now this is the clear. And even I request you to adopt some project in your college subject or in the curriculum to take some project or to do some project on the separation or the bioseparation techniques also you can use. Some biofilm you can use for the separation. Okay, so the technique biorefineries are nowadays is coming that concept. So this type of, because I am just giving you the keywords. Further, you can search that types, or you can communicate with me after this subject also, training also. So as a teacher, definitely I will help you as much as possible because your mind should explore the new and new area. Okay, so now stage 5, the product purification. So that is one of the important issues because whatever product we are producing, the market only will accept with its quality or the grade. That means with a particular purity. Sometimes purity may not be very important. Sometimes purity is very much important. So as I already told you, sometimes it is possible, sometimes there are some market for the 100% alcohol, but sometimes it is for 8%, 42%, 80%, 90%. So as per the market demand, we will go through the purification, and product purification is nothing but a separation technique. Am I right? So, but that separation technique is the precision separation. So high performance separation technique is required for the purification. Product separation and product purification both are the separation techniques, but product purification word come to our mind and application of precision making separation technique or high performance modern separation technique. And product separation, because we have to deal larger amount of material. So sometimes it is not possible to use the modern membrane because large amount of large components are there. Okay. So after that, product storage. Storage is also very important factor, because whatever product you produce, you know that that all these products, whether it is the herbal products or the chemical products, all there, or the medicines, whatever there, what happens? So we have to purchase it by checking its expiry date. Okay, checking it. Okay, Samyukta, I already told you that high amount of product separation we can do with the distillations, evaporations, extractions, filtrations, all that, and precision making purifications, as I say, the chromatographic separation, liquid chromatography separation, par evaporation, membrane separation techniques like that. Okay. Anyway, the product storage. You know, what is the first job while you purchase medicines or you purchase herbal any products from the market? What thing we have to check whenever we purchase anything from the market? What I have to check first, guys? Yeah, expiry date. Why? And why? Yes, expiry date, Nancy. Why? Because in our mind it is working that no products, almost no products maintain its quality or maintain its grade independent of time. So unlimited stability of the products is not possible. So okay, it might be toxic, because that means toxicity why appears? Because of the degradations, because of the, it is called the weathering effect of weather. If some leakages are there, and even complete packaging is there, no exposure of air, weather, still it degrades. That means stability of the products is an important issue. And the stability issue is not only for the products, food products or the usable products. It is also true for the petroleum or diesel we are using it. You can't store the diesel with a closed container for the long time and use it and get the same performance. No, because that also have some problem. Okay, so that expiry date. That means product stabilization. So product storage, I told you initially, guys, those are working, those people are expertise in the storage and packaging, and one of you are working in the reactor, and one of you are working in the packaging. So no one is superior and no one is inferior. So every sector, every stages, we have a challenge. We have to face challenge. It's a big issue. The stability of the product, you know, we have to think about it. And sometimes we give some preservatives, some packaging techniques. So sometimes it is required to have anoxic, so inert environment for the storing. So the different storage techniques or strategy we have to find. So then market. you can sell it. So I hope you are not able to give any example without this all these stages I have mentioned in the anatomy of chemical engineering. Now I hope you can think a refinery, what are the steps of there? If fertilizer, what are the steps of there? A distillery unit, what are the steps of there? All these steps are there. In a petrochemical industries, yes, all these steps are there. So this is the anatomy as you find the anatomy of your of the animal body or that other, the backbone. So this is the steps, and we say in engineering language, all are units. Unit one, unit two, unit three. There are different units in the industry. So if someone asks you, do you have idea about the units in the chemical plants like refinery? I hope now you can take part in that debate. Is that clear, guys? I hope now you get the confidence that you get. Okay, so you will be able to say that these are the steps. Whenever any employment, anyone asks you the question, I hope you can answer. At least, initially you don't be confused and nervous. So, and it's unknown to you, you will be able to answer, right? Now, gradually I will discuss or enter into the depth of the topic. So, but my intention is that whatever you learn, let it be very clear in your mind so that you will not forget it. Okay. So,